Welcome back. So we've started looking at primary structure, and we said that this is simply the sequence of amino acids which are joined by peptide bonds. And that sequence is specified by the instructions that are contained in a gene. Instructions that are contained in a gene. So remember, the other groups distinguishes amino acids from each other or from one another. So they play no role in the peptide backbone of proteins. And hence a protein can consist of any sequence of amino acids. So a protein can consist of any sequence of amino acids. So remember, we have 20 standard amino acids. You have 20 types of amino acids that are available. And the length range from 2 amino acid residues, which is a dipeptide. And the number of protein structures that are possible, hence, is actually practically limitless. So in fact, there actually may be 10 to the power n different combinations of amino acid residues for a polymer, the given length of n. So a polymer with a given length of n, for example, if a peptide is just 10 amino acids, it means that there are about 20 to the power 10 possible sequences, possible amino acid sequences. And henceforth, the sequences that can be formed by amino acids is actually limitless. Now, why is the order and type of residues in the primary structure of a protein important? So remember, we said that R groups present on each amino acid will affect its chemical reactivity and also its solubility. Therefore, we are able to predict that the R groups that are present in a polypeptide will also affect that molecule's properties and also its function. Now we find that in some cases, that even a single change in a sequence of amino acids will actually cause a radical change or radical changes in the way the protein as a whole behaves. So for example, a single change in hemoglobin, HB hemoglobin, is linked to a disease of sickle cell. I'm sure you may have heard sickle cell before, or someone who has sickle cell. So just a change in position 6, just a change in position 6 here, a change where this glutamate has been replaced by valine, brings radical and drastic changes in hemoglobin. It is going to lead to the red blood cells to change from their normal disc shape to a sickled shape. And hence, that's where the name sickle cell is derived from. So just a single change brings drastic results. Remember, each and every amino acid has their properties. They have different properties. They have different properties. So in this case, for example, we have glutamate that has been replaced by valine. So where there was glutamate, there's valine. What type of amino acid is glutamate and what type of amino acid is valine? Guys, what do you think? This is a recap. It's another recap. What did we say? What type of amino acid is glutamate? And what type of amino acid is valine? So remember I said glutamate has a negative charge on the side chain. So this is, so what type of amino acid is this? Guys, glutamate has a negative charge on the side chain. What amino acid type is it? Guys, anyone, or maybe I'm speaking to myself?
You are not, sir.
Yes, what type of amino acid is glutamate? It has a negative charge on the side chain.
I think it's acidic.
Yes, it's acidic because it has lost a proton when it has a negative charge. It has lost a proton. So it's acidic. Whilst valine is non-polar. It has a non-polar side chain. So remember, an acidic amino acid has been replaced by a non-polar amino acid. I remember glutamate is actually polar as well. So you have a non-polar being replaced by an acid. Glutamate, which is acidic, has been replaced by valine, which is non-polar. So this is simply the primary structure. It's a sequence of amino acids, simply a sequence of amino acid, sequence of amino acid. What primary structure is this? What molecule has this primary structure? 1 up to 29. Okay, find out in the next class. We'll talk about this. So the importance of the primary structure, it is fundamental to the function of the primary, or to the function of the protein itself. And then it is also important for the higher levels of protein structure. It's very cardinal for the secondary, tertiary, and quaternary structure and quaternary structure. Okay, so that's the primary structure. Now for the second structure, this is the next structure after the primary structure and is created in part by hydrogen bonding between components of the peptide bonded backbone. So of importance in the secondary structure is or are hydrogen bonds. So the secondary structures are actually distinctively shaped sections of proteins which are stabilized largely by hydrogen bonding, which occur between oxygen on the carboxyl group or the C carbon covalently double bonded to oxygen group of one amino acid residue and the hydrogen on the NH groups of another. So forms between oxygen on the carbonyl group of one amino acid residue and the hydrogen on the NH groups of another. Why does this formation happen? So we understand that the oxygen in the carboxyl or carbonyl group, rather, the C double bonded to O group has a partial negative charge. That oxygen has a partial negative charge because it has a high electronegativity. It has a high electronegativity. While the hydrogen atom in the NH group has a partial positive charge because it is bonded to nitrogen. And nitrogen has a high electronegativity. So no wonder we find that there's that formation of hydrogen bonds between the carbonyl group and the hydrogen atom from the NH group. So the oxygen has a high electronegativity, and hence it has a partial negative charge. The hydrogen, the oxygen has a partial negative charge due to its high electronegativity. The hydrogen atom in the NH group has a partial positive charge because it is bonded to nitrogen, which has a high electronegativity. So no wonder we have that formation. Now of note is that the hydrogen bonding that occurs in the secondary structure between the sections of the same backbone is only possible when these polypeptide bonds are in a way that are going to put the CO group, the carbonyl group, and the NH groups close together so that they're able to form those hydrogen bonds. So they need to be close together. So when these polar bonds are actually aligned and form the hydrogen bonds with one another, these happen when the backbone bends to form one or rather one of the possible structures. So they can either form what is called the alpha helix or the beta pleated sheet. The alpha helix or the beta pleated sheet. So this is the alpha helix. Alpha helix. So the alpha helix is actually coiled. It's coiled. And then when you look at, we also have the beta pleated sheet. So this is the beta pleated sheet. This is the alpha helix structure. So proteins are able to assume any of these two structures. Are able to get any of these two structures, either alpha helix or beta helix. So in both structures, you're going to find that the distance between the residues that the hydrogen bond to one another is actually small. So for example, in an alpha helix, the hydrogen bonds form between residues that are just 4 linear positions apart in the polypeptide sequence. In the alpha helix, they are going to form between residues that are just 4 linear positions apart, 4 linear positions apart. So they are apart, just 4 linear residues forming the alpha hydrogen bonds. So now, which one forms either the alpha or beta structures would depend on the molecule's primary structure. So specifically, we're talking about the geometry and the properties of the amino acids in the sequence are going to determine which structure, whether it's alpha or beta. We find that certain amino acids are actually more likely to be involved in alpha helices than in beta pleated sheets, and vice versa, due to the specific geometry of their side chains. So the side chains are very important. For example, if I can give an example of the special amino acid, proline, it may be present in beta pleated sheets, but when it is found or present in alpha, when it is found in alpha helix, this is going to terminate the alpha helix due to its unusual side chain. Remember, I said that the proline causes a kink in the amino acid chain. So the amino acids are going to determine the structure to be taken by the sequence, whether it's alpha helix or beta pleated. And I've given an example of the proline because of its R group. So the bond that is going to be formed between the proline's side chain, the R group, and the nitrogen of the core amino acid group is going to introduce kinks in the amino acid backbone or in the peptide backbone, which do not conform to the shape of the helix. So no wonder the formation of the side chains in a protein sequence or in an amino acid sequence is very important. Now, the tertiary structure of the tertiary structure. This is the overall shape that is assumed by each peptide or by each individual polypeptide chain. So remember, this final folding of a protein is actually determined by its primary structure, the nature of its side groups. Now, there are various forces and interactions that are going to stabilize the secondary structure. That is going to stabilize the secondary structure. There are a number of forces or interactions in a tertiary structure. So one of the forces that act in the tertiary structure, so there are actually 5. So one of the forces are the hydrogen bonds. The hydrogen bonds. So these hydrogen bonds are going to form between polar side chains, polar arrow groups, and opposite partial charges. Opposite partial charges. Opposite partial charges. Either in the peptide backbone or other arrow groups. So you have the hydrogen bonding, of the hydrogen bonding taking place there. So between the polar arrow groups and opposite partial charges, hydrogen bonding. The other one that takes place is a hydrophobic interaction. The hydrophobic interaction. So find that in an aqueous solution, water molecules are going to interact with the hydrophilic polar side chains of a polypeptide. So when it is in solution, in aqueous solution, the water is going to interact with the hydrophilic part or portion of the polypeptide, and hence force the hydrophobic non-polar side chains to coerce into globular masses. So it's going to interact with the hydrophilic part, and this is going to force the hydrophobic portions, these, to coerce into globular masses. So now, when these non-polar arrow groups come together, we find that the surrounding water molecules are going to form more hydrogen bonds. They're going to form more hydrogen bonds. Hence, this is going to increase the stability of their own interaction. So you have hydrogen bonds, you have hydrophobic interactions. The other interactions are the ionic bonding. So these are going to form between groups that have full and opposite charges, such as ionized, acidic, and basic side chains. So they are going to form between opposing charges. So you have the negative charge there, and you have the positive charge there, forming an ionic bond. The other bond are the covalent bond. The covalent bond. So these are going to form between the side chains of 2 cysteine through a reaction between the sulfur-hydryl groups, sulfur-hydryl functional groups. There will be a reaction. So 2 cysteine. Cysteine is an amino acid. It's a sulfur-containing amino acid. So remember, we have 2 sulfur-containing amino acids of importance that you need to know. We have 2 sulfur-containing amino acids. We have methionine, it's a sulfur-containing amino acid, and also cysteine. So these covalent bonds of the disulfide bridge or bond is going to form between 2 cysteine amino acids through a reaction between the sulfur-hydryl functional groups. So this is a bridge, forming that bridge. So this interaction, this bonding, create a strong link. It creates a strong link between distinct regions of the same polypeptide, or even 2 separate polypeptides. So it's a very important link or bond, because it actually locks particular regions together, the disulfide covalent bonding locks particular regions together. Then the last one is the van der Waals interactions. The last interaction that takes place is the van der Waals interaction. The van der Waals interaction. So remember, we have the hydrophobic side chain. So if once those hydrophobic side chains are close to one another, their association is actually further stabilized by electrical attractions that are known as van der Waals. Now, the van der Waals interactions in and of themselves are actually weak. These interactions are weak. And they occur because of the constant motion of electrons, which gives the molecule a tiny asymmetry in the charge that changes with time. So that charge, asymmetric electron, that charge, that constant motion of electrons gives or yields to these van der Waals interactions. So even though these van der Waals interactions are weak relatively to the covalent bonds, the disulfide bonds, or even hydrogen bonds, we find that if a large number of van der Waals attractions are present, this significantly is going to increase the stability of the structure. So there are 5 types of interactions that are involved in the tertiary structure. Okay, so after the tertiary structure, we are supposed to look at the quaternary structure, structure. So let's look at a summary of what we've looked at today. So the key thing of us to note is that protein structures, there's a hierarchy in the protein structure. So the quaternary structure, which we are going to look at further the next time we meet, is based on the tertiary structure, which is also based in part on the secondary structure. So find that all these higher levels, all the 3 higher levels, the secondary, tertiary, and the quaternary structure are based on the primary structure. Then the combination of primary, secondary, tertiary, and quaternary levels of structure is actually responsible for the fantastic diversity of sizes and shapes that we observe in proteins. And then protein folding is directed by the sequence of amino acids that are present in the primary structure. And then most elements of protein structure are based on folding of polypeptide chains. They are based on the folding of polypeptide chains. So does protein folding occur spontaneously? What happens to the function of a protein if normal folding is disrupted? What happens to the function of a protein if normal folding is disrupted? So we're going to find out next time when we meet. We're going to find out the next time that we meet. So do we have any questions? So next time we'll meet, we're going to look at the quaternary structure and also look at some specific examples of proteins or peripeptides, and learn some things from there. So we'll start from quaternary structure, and then we'll look at specific examples of important proteins or peripeptide chains and apply what we've learned today.